G'day viewers, I'm here with Ken Hayworth, neuroscientist extraordinaire, who's currently researching at the Howard Hughes Medical Institute and is the president of the Brain Preservation Foundation. And that's where John Smart also does some stuff for them too. So I'm just wondering, Ken, can you give us some background on yourself and your history?
Sure. Thanks, Adam. My principal background is as an undergraduate, I did computer science and engineering. In graduate school, I have a PhD in neuroscience with a concentration in cognitive science, and I did a bunch of research while I was in the neuroscience PhD program on how the human visual system recognizes objects, and specifically the relations between the parts of objects. At the same time I was doing that, I invented a little thing called the tape collection mechanism for ultramicrotomes. I did that with Jeff Lichtman at Harvard University. I was kind of flying back and forth between Los Angeles, where I was getting my PhD, and Harvard University in Boston to do that work. And then after I got my PhD, I did a postdoc in the Lichtman lab that really continued that work. Which got to a certain level of completion, because right now that tape collection mechanism for ultramicrotomes is being licensed as a commercial product, and hopefully it will show up in a bunch of neuroscience labs pretty soon. I know a few that already have them.
Fantastic.
Then after that postdoc, I went to the Howard Hughes Medical Institute Genelia Farm Research Campus, which is just the most awesome place for doing connectomics in neuroscience that there is in the world today. I am very pleased to be able to work on a project whose goal is to take an entire fruit fly brain and map it at a resolution of 8 by 8 by 8 nanometers. So the entire fly brain at 8 by 8 by 8 nanometers, using a technology called focused ion beam scanning electron microscopy. That is not something I invented. That's been around for a while, but I am putting a bunch of improvements on it with my colleagues at HHMI to try to get to the level where we can do a whole fly brain, and hopefully pretty soon even beyond that. I'm hoping to scale that particular technology to arbitrarily large volumes.
Fantastic. Oh, look, your tape collection mechanism for ultra micro, ultra microtomes, is that correct? Yep.
Yes.
Is that similar in nature to the molecular ticker tape technology that
Uh,
yeah, I'll let you.
yeah. No. No. It is definitely much lower tech. So. So the traditional way that people do that people have been studying the connectivity of the nervous system, the circuitry of the nervous system, is to use something called transmission electron microscopy. And so back in the 1950s, there was a fantastic set of inventions, one of which was the newly invented transmission electron microscope, and the other was an ultramicrotome with a diamond knife. Sometimes they used glass knives back then, that could cut a plastic embedded piece of brain tissue at 50 nanometer increments, into 50 nanometer slices. And that was thin enough so that you could put them in a transmission electron microscope and see the profiles of the neurons in each subsequent section that had been cut. And so starting in the 1950s, people used the ultramicrotome and the transmission electron microscope to start really looking at the three-dimensional structures of the way neurons connect in the brain, and to really understand what is happening there. So that's the ultramicrotome. That technique has never really been automated, but within the last 5 to 10 years, there have been 3 techniques that have come along that have modified that to a great extent, that really allow much more automation in the process of mapping neural circuits. And these techniques are really the thing that is driving this field of connectomics, this idea that we can get all the connectivity in a large piece of brain tissue. And so there's 3 technologies that do that. One of which is this automatic tape collection mechanism, which is a very simple technique. It basically just takes the existing ultramicrotome that people have, that they cut 50 nanometer sections on for looking into it in a TEM. And instead, it puts a conveyor belt, if you will, into the mechanism so that it can collect thousands of these sections automatically without any human intervention. The trick is that these sections are collected onto a solid tape, so they can't really be imaged in a transmission electron microscope. They're imaged in a scanning electron microscope. But you can get just as good images off of them and trace the neural circuitry. There's 2 other techniques, one of which is the one I'm working on right now, which is focused ion beam SEM. And you'll probably ask me to describe that a little bit later. Anyway, the one that really kicked it off, this kind of revolution in connectomics, was something called the serial block face SEM, invented by Winfried Dink in Germany. And he should get the credit for really kicking off the revolution in connectomics that we're now within. Because he invented this, it had actually been suggested a little bit before he did, but he really brought this into a usable instrument. It is a scanning electron microscope in which you put a block of plastic embedded brain tissue. But within that scanning electron microscope is a diamond knife that scrapes off a few tens of nanometers off the surface. And then that scanning electron microscope images that freshly revealed surface. And it just does this over and over and over again within a vacuum chamber. And so you get image after image after image without any human intervention. And it can work for a month straight and get an enormous quantity of data from a block of neural tissue.
Sounds very interesting, and it sounds very similar to what Todd Huffman is doing with 3Scan.
That's right. That's right. Except for the fact that so Todd's work only uses light microscopy. It is a kind of a laser scanning through the knife technique, and so its ultimate resolution is maybe about 1/2 a micron, whereas the resolution that people routinely get in these other techniques is about 100 times better than that. Now, we pay the price for that because Todd's 3Scan can go through an entire mouse brain in a reasonable amount of time, just a few days, I believe. Whereas if we were going to go through a whole mouse brain at 100 times the resolution, that would mean 100 times 100 times 100 times more data, more voxels in terms of volume. So 1,000,000 times more. It would take 1,000,000 times longer with our techniques, roughly. And so the real way to understand this is that Todd's technique can see an entire mouse brain, but it can't really see the circuitry. What connectomics research is trying to do is it's trying to see every single connection between every single neuron in the volume that it's looking at. And to do that, because the wires between neurons go down to about 40 nanometers in diameter, you have to use an electron microscope, and you end up having a much more difficult technique than light microscopy, and things that take much longer to acquire the data.
Right. Is this something that you think may improve in price performance in the near future, or is it like, is there physical limits which are being sort of scraped up against now, which are going to sort of retard the progress, or not retard the progress, but it will not make it as quick as something like Moore's law for CPUs?
Yeah, well, I'd have to say both. I know that was an either/or question, but
Yes.
uh, so so the current techniques definitely are scraping up against physical limitations. So, for instance, the imaging that I do in a scanning electron microscope is limited by the number of electrons that I can actually get to each pixel. And so I can only acquire at maybe 10 million pixels per second, and I can't really go any faster than that given the physics of the electron gun that I'm using that is available today. But when you talk about Moore's law, we are actually, connectomics is actually poised to piggyback on top of Moore's law. So the fundamental instrument that people use in connectomics is the scanning electron microscope. It is the fundamental instrument for doing any of these things, because what it is doing is it's firing a bunch of electrons into a very small spot and looking for the on a piece of brain tissue, and it's looking for the electrons that get backscattered. Now, that to scale that up to get larger and larger and larger volumes faster of neural tissue, what you really need is you need many of these electron beams very cheaply, imaging either the same surface or many machines in parallel, imaging blocks that have been cut off of a larger block. And coincidentally, this is exactly what the semiconductor industry needs as well. The semiconductor industry is reaching the limit, Moore's law is reaching the limit of light photolithography, photolithography that uses ultraviolet light. And it is trying to develop the techniques to use electron beams, E-beam lithography, to replace its basic manufacturing processes. And in the process of doing that, it has developed multi-beam electron microscopes and electron microscopes that can be built cheaply and mass produced. And so all that really needs to happen is that technology that's being pushed by tens of millions of dollars, hundreds of millions of dollars by the semiconductor industry, that technology needs to be transitioned for use in imaging neural tissue. And at that point, we would be thousands of times faster than we do today. And the scale of volume would not be a fly brain, it would be much larger. And this is not pie in the sky. There are at least two main connectomics labs that have now purchased multi-beam electron microscopes, 61 beams that are imaging all simultaneously on a piece of neural tissue. And they have very grand ambitions for using this to map large pieces of the mouse brain. And it's just the beginning.
Yeah, wow, that sounds very profound. What kind of time scale are we looking at where we can see these multi-beam microscopy like being used at not just experimentally to test the technology, but being used practically in labs and having practical results from tests that are using these instruments?
Yeah, well, the two labs that I'm talking of, they are definitely getting test instruments. but they have gotten these test instruments under grants that expect real scientific results.
Mm.
So if they meet their progress timelines, 3 years. And they should be reconstructing pieces of mouse retina, etc.
Right. So I mean that's a lot bigger than the fly brain. Uh, but like you mentioned, much larger brains or parts of the brain should be able to be scanned quite easily. How much larger?
That's right. Well, so largeness also plays off against resolution.
Hmm.
So the work at Harvard University in my old lab, they have a multi-beam that is targeted to reconstruct part of the mouse retina. And yes, it would be quite a large piece. Um, but it would be at a resolution that would be close to the best resolution that you need. It would be on the order of 5 nanometers or less pixel size. So these would be very high resolution images, and you could see everything within it, every little tiny process.
Mm.
The other group that is looking into this, which is Winfried Dink's group using his Sierra Blockface SEM, they are trying to do a whole mouse brain. But they are, at least for now, concentrating on doing a whole mouse brain at a lower resolution that is still stunningly higher resolution than what you could get with a light microscopy. For instance, with this would still be 1,000 times more data than Todd Huffman's 3-scan device, because it's using an electron microscope.
Sure.
This would be a whole mouse brain where you could trace every long-distance myelinated axon in the brain. Okay, and to give you an idea, this would not be all the tiny neural circuitry,
Yeah.
but
Sure.
it would allow you to see every single region of the mouse brain and how it topologically projects to every other single region of the mouse brain. It would, in one experiment, it would actually replace 1,000 tracer injection experiments that have been being done over the years. It would really be stunning, and they need a multi-beam. It sounds like, from what I've heard, to do that in a practical amount of time. But they're going to get it because they're piggybacking off of the advances in Moore's law in the semiconductor industry.
That's profound. So, I mean, it seems like there's over the last 10 years, there's been massive inroads into neuroscience from an outsider who's not a neuroscientist but very interested in this subject, looking at the sorts of technologies that seem to have I guess become available to the public eye, Brainbow, and which was very beautiful and captured people's imagination because it made all the neurons lovely colors, and people could see what was going on, and all the science magazines got on board and got these pictures of the Brainbow things and put them on the front covers, and then people started getting exciting about the brain again. And then the optogenetics and I mentioned something before. What was it called? The molecular ticker tape and things like that. It seems to and the resolution of MRIs are getting better, but that's really just measuring a lot of the metabolism in the brain, not the actual what's going on really. So yeah, this sort of technology, the multi-scan, the multi-beam technology seems to be quite profound.
Yeah, I mean, I mean, neuroscience has definitely been experiencing some fantastic advances. You could probably put those into two categories: the ability to measure function directly of large numbers of neurons, and the ability to map out the connectivity of the neural circuits that that give rise to that function.
Hmm.
And I guess I should add a third one because you mentioned optogenetics, the ability to manipulate that activity and see what happens. And so these are the intersection of these is really, really exciting because now you have the ability
Exactly.
to look at calcium sensitive dyes so that you are watching hundreds of neurons light up in whatever pattern they need to light up at in an awake behaving animal, like a zebrafish or a fly or a mouse visual cortex. And then you're able to see those neurons responding, not just a single one with an electrode, which is what has been being done for decades, but a whole field of these neurons responding to a wide range of stimuli and behaviors, etc. And then to take that same animal and perfuse its body with fixatives and remove its brain and remove that little chunk and slice that up and put that under the electron microscope, and then trace out the neural circuitry that actually gave rise to that behavior. This is something that neuroscientists a decade ago could only dream of,
Mm.
and now we're doing it. And if you have, I come from an electrical engineering background
Hmm.
to a certain extent,
Mm.
and I wouldn't know what to do without an oscilloscope. I mean, if you want to know how a circuit works, if you want to really get into it, you need the circuit diagram.
Mm.
You
I was
need
going to
to
bring up
have
that. Yeah.
an understanding of what the transistors and the resistors and the capacitors are and all their labels, and you need to get in there with an oscilloscope and some measurement instruments and see the thing, how the voltages and currents are actually moving around in the circuit. And if you can do that, you can understand the circuit. And there really isn't a difference between an electrical man-made circuit like that we would be trying to figure out, and the brain, except for the fact that our instruments were just not good enough. I mean, obviously they behave differently, they're built differently, but they're both machines, and they're both processing information with little pulses that are being transmitted from one component to another component. And so, having this ability to see the components in operation, to see the signals in operation during behavior, and then to get the circuit diagram, uh, it changes the field of neuroscience. It really
Yeah.
becomes
That's
something that is much less vague theorizing, uh, trying to figure out this black box that we really can't open up, and being able now to just open up the black box and say, okay, here's everything that could be causing this behavior. Let's see it in action and dissect it.
I look. That's just incredible. I mean, like understanding a brain like a circuit diagram is like understanding how circuits work. And if we understand how circuits work, then we can bottom up understand how our computers work, right?
Yes.
Except we're not just understanding the desktop PC or understanding, the supercomputer or whatnot. We're understanding what's going on in here. And this is incredible. I mean, like human beings have been philosophizing about how the brain or how the mind works ever since recorded history. And there's been so many theories based on vague concepts that some have been ridiculously wrong, like the whole uh what was it called, the 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 blood bile, the yellow bile, and like the black bile, the sort of, what was it called? It was the
I don't know. They don't teach us that in school anymore.
no, 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 they don't teach that anymore. But like it was a theory of mine that was based around certain forces being propelled around the body. Yeah, so it just escapes me at the moment. But it is crazy how far we've come, and it looks as though we're going through some sort of new renaissance in neuroscience. Um, it's like,
Yeah,
yeah,
yeah.
sorry, I was
Yeah,
going
I
to
was going
say
to say.
something about the circuitry understanding the brain and the circuitry, and the neuron's kind of like an RC sort of circuit, isn't it?
Yeah. Well, so so neuroscience is fantastic. The understanding of how a single synapse works and how a single neuron works is at such a level that we can, and this has been going on for at least 20 years, we can simulate neurons uh, at the level that really gets to their behavior. There are great neural simulators like there's one called Genesis that's been out there for quite a while that simulates neurons as compartmental models with ion channels that are opening and closing, voltages, all of the stuff that really matters, just like an RC circuit. In fact, it is an RC circuit if you look at the way these models are put together. But that's and those models they are coming together with the connectomics that actually show the larger wiring of pieces of brain. It's a great story, but at the same time, I don't want to give the idea that that connectomics is really the only thing that is pushing forward understanding in the brain, because the truth of the matter is connectomics has hardly told us anything yet about how the brain works. It is developing tools mostly right now. It has provided some interesting clues, but there has the last 50 years in neuroscience has fantastically pushed forward our understanding of how the brain works. And that's just neuroscience. If you look at cognitive science, that it is coming up with very, very good models of exactly what you were saying, of what is happening in here from a computational perspective. What are the algorithms that are going on? So if you were to ask me, and I'm sure you won't ask me all these things, but if you were to ask me, okay, how does somebody recognize an object? How does somebody recall a memory? How does somebody learn how to learn high school algebra from teachers' examples in natural language? These are things that are being modeled with cognitive models that are very they are very well supported in experimentation. These are very good theories about how the brain actually does these complex tasks, and so we really are as a field of neuroscience and cognitive science picking apart this 3-pound brain within us. And so we really do understand much more than a lot of people think we understand about how the brain works already.
Hmm. That's crazy. It's amazing. Look, there's got to be some like a. Well, let's have a look at flight. I mean, we tried flying for a long time until like the Wright brothers came along and actually did it, right?
Hmm.
And so they didn't just come up with all their ideas by themselves. I mean, they were standing on the shoulders of giants. But as soon as flight was achieved, um, by the Wright brothers at the I think 1905 or 4 or something or 7 not all that long ago, about 100 years ago. 60 years later there are footprints on the moon. Okay.
Yeah.
um, so connectomics, if it hasn't actually given neuroscience anything new, it's perhaps because it hasn't reached that tipping point. And if that tipping point is close, like you were suggesting before, um, then maybe we will see some like a massive influx of new information about the brain that hasn't already been obtained via classical means. And before, because I'm silly and I always forget stuff, I was talking about humorism when I was talking about the black, red, and spitball. This is like the old way that people used to think, intelligence arose from. Yeah, you
Mhm.
aware of that? The sort of
Yeah, yeah. I can think of some other crazy theories as well that were rolling around back then.
Yeah, yeah, yeah. So what do you think is the most funny, crazy theory rolling around about how the mind worked
Uh, I have to say, I mean, the 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 craziest theory about how this probably is not answering your question,
hmm
okay?
hmm
But the theory that 99% of our fellow human beings has about how
today?
the mind works is actually that the mind is not a machine at all, that it doesn't have pieces. I mean, if you I was listening to a YouTube video from Sam Harris,
Yeah,
one of my favorite atheists,
yeah.
and
He's great. Yeah, the moral
he's
landscape.
fantastic. And he was saying, I think this really cuts to the core. He was saying, um, he was saying if we damage a part, if we damage our eyes, we go blind. If we damage our part of our brain, we lose the ability to speak. If we damage another part of the brain, we lose the ability to have any of our memories. And you went through a whole list like this. And then he said, but uh, what religious people think is that if you destroy the entire thing, then somehow miraculously all these things come back. So why do we need eyes to see if our soul can see? Why do we need a brain to remember things if our soul does the remembering? And so this idea that intelligence and consciousness uh, is is um, is a uniform substance that is uh, indivisible, is homogeneous, that it is unique in each of us, and that somehow it just got this body and will go on after this body is destroyed. That's the theory that's the absolutely craziest out there, and it's still haunting us to this day. Neuroscience has not done a good job of convincing the public, if you will, that we really are machines and that we're figuring out how we work.
Yeah, yeah. It's crazy. Well, I mean, like, I guess the way that we've evolved has made us sort of inquire about things and think about things from in a certain way. I'm not sure if this is correct, but maybe if we weren't taught anything, if we didn't have the sort of access to the sum of knowledge, um, a lot of us would grow up thinking along the lines of some of the early animists, and like we'd project a sense of self onto our environment, and perhaps my theory is that because of that, it's been we've evolved to sort of take advantage of that way of thinking over the hundreds and hundreds of thousand years of our ancestry surviving in the environment and ascribing some form of agency to a dangerous object or to predators in it as though they had like a similar agency to us was a way that we'd sort of survive, but also as we got smarter, and it only after a certain period of time did we start, I guess, doing burial rites and asking and perhaps asking questions about the afterlife or what happens when we die. We never used to think about that more than 200 or 150,000 years ago, somewhere around there. We started thinking about those things relatively not all that long ago, according to historians or some yeah. So it's interesting to think that we picked that up, but maybe that came from our previous animistic way of looking at the world. I don't know for sure. That's just a theory.
Yeah,
But
no, it makes it makes a lot of sense.
hmm,
And I guess the main point that I would like to get across on that is that uh, this theory, we might like to think, oh, we've grown beyond this theory. But the minute that okay, so I'm as you know, a vocal advocate for mind uploading eventually happening,
Mm.
and for us getting there through preserving our brains and eventually mapping our brains maybe 100 years from now when the technology is perfectly well developed, and coming back as uploaded beings in machines. And the way to see that this animistic thinking is still with us is to look at the scientists and even neuroscientists and skeptics that look at that idea and they say, well, that would just be a copy of you. It
Right.
wouldn't really be you. And I've seen some really great skeptics that say that, you know,
Mm.
and without any thought really to be
Mm.
giving. Okay, so you're saying you've copied all of the things that neuroscience says is us, and yet that copy is just a copy.
Mm. Just a copy.
What's missing? The special stuff, the special sauce that's not made of atoms. Is that the stuff that's missing? Um, so it is very pervasive, and I think uh, the I think that that's actually killing us. I think that our inability to truly embrace what science has told us about the mind and about the brain. The
Mm-hmm.
brain is a machine. The mind is software that's running on that machine,
Mm.
and software can be copied, and it can be run on a different substrate. And it is so obvious to me that that's what neuroscience says. That's what cognitive science says. I would have to bend over backwards to find anything remotely in all my neuroscience, all my cognitive science textbooks that I've read over the years, to see anything that even comes close to being against that theory. And yet, if you really accept that theory, then that means that the most important technology that we can be doing today is figuring out how to preserve a brain. This is a technology that is within our grasp. It is so well defined as in terms of a clear goal, in terms of clear obstacles that we need to and clear research that we need to do, and yet there is essentially uh, almost no one in the world working on this technology, and yet this is the technology that can cure every disease that human beings have, and it could achieve it within 5 years if we just put in the research effort, and yet it is not being done. And if you ask why is it not being done, it always comes back to the same point. They we really don't believe that we're machines. We really don't believe that we're software, that our minds are software. We think that there's some magic stuff. And I put the man on the street who's carrying his Bible that
Mm-hmm.
would say that. I put him along with the neuroscientist that would say, oh yeah, but there's some magic in the biology that you're going to be missing. I'd put it along with the skeptics that say this is it's just a copy. There is at the core of all of these arguments against why we should be looking into brain preservation is this animistic soul idea that is still lingering, and we have to exercise it, if you will.
Yes, I get that. So, it's interesting that you say like you could cure a whole lot of diseases. Now, I can think through the logical possibilities of curing a disease by uploading. or by yet some or simulating the mind in a sense. The simulated mind would be sort of abstracted away from the bodily conditions, and you'd therefore be able to have you'd have the tinkering kit, the engineering kit, to engineer away anything that would be considered dis-ease or some form of suffering, maybe. But I'd like to hear you explain that, because maybe not a lot of people would be on board with that idea.
Yeah. So yeah, let me lay out this in very concrete terms. Uh, so we know that the brain. So what is neuroscience telling us? Neuroscience is telling us that that all of our memories, all of our all of our skills, all of our conscious experience, they are coming from the brain, and they are encoded in the circuitry of the brain in the way that the 100 billion neurons are connected to each other. Okay, and as a neuroscientist and as a cognitive scientist, I could go into great depth in any area of the brain and tell you the textbook theories on how we recognize objects, how we form memories, how we make decisions, and I would be pointing to collections of neurons and how they are connected structurally together. We know that this is the basis of human identity. Now, we don't have any ability right now to correct a brain disease or to correct a bodily disease that that is beyond medical science. Obviously, there's no easy cures. If somebody has incurable cancer, there's no easy cure by definition. But the stuff that makes that person unique, the memories and the skills and the personality traits, all of those things that make that person unique are encoded in this structure of the brain. And we know for animals, for small animals' brains, we know how to preserve those synaptic connections. We know how to do it. And for small animal brains, we know not only how to preserve those, but we, with the connectomics technologies that I told you about earlier, we know how to read those off. We know how to read the circuitry off. We know how to figure out where every synapse is connecting. And actually the strength of those connections written in the structure of the of the sizes and of the synapses. And so we have these technologies for small scales to both preserve all of that synaptic connections and to read those off. And so the only thing that you have to really think of is, as that technology progresses, we will eventually get to the point where we can preserve an entire human brain. Okay? Now, I would argue, and I can give you some evidence for this later, I can argue that that is not a very difficult problem. It is a research problem that we can't do immediately, but it is definitely not a very difficult problem on the scale of things. Now, in order to read off that circuitry, that is a very difficult problem. It is a difficult problem in the same way as going to the moon was a difficult problem. It is an engineering feat that that is well beyond our current engineering capability. But at the same time, we know the tools, that it's just scaling up that needs to be done. It's just as in quotes, but it's just scaling up. So given $100 billion, we could take a human being and map the entire brain. I have no doubt of that. Given that kind of budget and a few decades time to do it, we could map an entire human brain. Do we have the willpower to do it? Probably not. We don't have the willpower to go back to the moon, so why should we have the willpower to do that? But we can see a path to getting there. And so the concrete thing is you preserve a brain so that all of these synaptic connections between neurons are preserved in a static state. That stays on a shelf for 50 years, 100 years, whatever is necessary, so that the technology to read it off gets developed and gets made reliable and cheap enough to actually apply it to human beings. And then once that circuitry is read off, we will understand how to simulate that. We already have a reasonably good idea of what to look for. We will understand within that type of time scale how to simulate that brain. And once you're able to simulate that brain and hook it up to a robotic or virtual body and turn that simulation on, what you should get is, if you do it right, is you should get the person back. They should open their eyes, and they should they will know that they are themselves. How will they know that they are themselves? The same way that I know I'm myself when I wake up in the morning, which is that I look at my, I recall episodic memories within my memory systems, and say, and reorient myself with the world when I'm waking up. And there are people that have these systems that are broken, and lo and behold, they wake up and they think that this is the first moment that they've been alive, and they don't know who they are. And so the idea of curing disease, obviously it's not a cure that allows somebody to go into a hospital, get their horrible, debilitating disease cured, and then go back home that day or later that week and be with their family. No, we don't have the ability to do that. And I unfortunately are not going to have that ability to cure all diseases like that for a long time. But are we going to have the ability for somebody to go into the hospital and the doctors at the hospital say, look, we can't do anything for you right now. You're going to die. There is no way that we can prevent that, but we can preserve your brain. And that gives you a chance at coming back 50 or 100 years from now. And I think that is a very realistic scenario. People just need to devote the proper resources to it.
Wow, that's amazing. So, there's a couple of questions I was thinking of while you were speaking. One of them was just a quick sort of question about what you about your idea of John Searle's Chinese room argument,
Yeah.
and the next question was how does the Brain Preservation Foundation, the plastination sort of technique, compare to the normal cryonics or the popular sort of cryonics way of doing things, of preserving brains? So first, let's start off with the John Searle argument. How would you respond to, let's say John Searle was debating you right now, and he says, but we've got this dude in a room who's like looking up like a symbolic dictionary, and the room is completely mechanical. It just doesn't equate to consciousness. We are a lot more than like some sort of homunculus in a room sitting there with a dictionary. Actually, he doesn't even say that the person doing the dictionary lookup is a homunculus. But yeah, I would like to ask you.
Yeah.
Well,
So,
maybe
so
do you think we could outsource our homunculus to China or something to take away some of the more menial tasks and let our real mind focus on more of the interesting aspects of creativity and intelligence? That's just a diversion. Never mind. Answer the first question.
yeah. So so I've read a lot about the consciousness question. I don't. In my research, it is almost useless, I suppose, but I am fascinated by the question of consciousness, and I am uh, I think the best person that has addressed those questions the clearest
Hmm.
is Daniel Dennett.
I knew you
Daniel
were going to say that,
Dennett
and
is
I
it?
do agree.
You knew I
Yeah.
was going to say, okay.
Well, I didn't know that, but I was 90% sure.
Yeah, so Daniel Dennett is very clear
Mm,
on these aspects. He handles the Chinese room thought experiment and others very clearly. Uh, in essence, the the cognitive scientists have already answered those questions. There are, if you are developing a system that a robotic system that is supposed to behave intelligently in the world,
mm,
then there are certain aspects of its program that that you can give it uh, that will start to look like what we call consciousness. And those aspects of the program are self models. And so the core idea of consciousness from a cognitive science perspective is it is has to do with our brain having a model of its own perceptions, its own actions, its own beliefs, and it maintains that self model, and that is what gives us our illusion, if you will, of consciousness. Of course, it's not an illusion, but that is the seat of our consciousness, that self model. So Daniel Dennett tackles that question very clearly. Uh, the same question has been tackled from a quite different perspective, but they're talking about the same thing. Thomas Metzinger, who wrote a couple of great books. I'm looking at one now, "Being No One," uh, and "The Ego Tunnel." These
I've
are
heard of
these
that.
are great books that really get into what it is about this self model that makes it seem to us like it has all the properties of consciousnus. And so they're answers to this. The Chinese room thought experiment, which you know, it was generated a long time ago, but um, it is a simple misdirection. The real question is uh, we know the aspects of the brain that the aspects of the cognitive processes in the brain that give rise to the behavior of consciousness, the behavioral outputs of consciousness uh, that we act like we have memories of ourselves. We act like that we have beliefs that we created a certain action, that we are seeing something, that we are in pain, or all of these things. And the real scientific explanation uh, lies uh, in picking apart that self model and how it is built into the brain. And I don't have time to go into it, but there's lots of fascinating things in that we've discovered in that direction as well, especially
Mm,
from the neurology patient literature.
yeah, right. Okay. So let's talk then about the Brain Preservation Foundation, and I think it's important because you're like director, and I've spoken to John Smart about this briefly before too, and there seems to be like some things that some of the mainstream clinicians disagree with about the Brain Preservation Foundation's approach, plastination approach. Would you like to just describe what the Preservation Foundation project is and what its major goals are, and the plastination approach?
Yes. Let me first of all correct something.
Okay,
There is no Brain Preservation Foundation approach.
right. Okay, whoops.
We are not uh, picking sides on this issue.
Right.
Okay,
Okay. Yeah. Sure.
and it
Well.
is there's a very clear reason why we're not picking sides. The cryonics approach has not been demonstrated to work. The chemical on a human being. The chemical plastination side has not been demonstrated to work on a human being. So how could we say that one is better than the other if both of them don't work? So the Brain Preservation Foundation is its main mission is to advocate more research and more mainstream research into any type of brain preservation. And to tell you the truth, it started with in my mind, at least, okay, I came up with the idea. It started with a much more highly focused on cryonics. Because after all, cryonics has had a longer history of attempting to do human patients, and being very explicit about its goals of doing a whole human patient and preserving the brain. So I actually thought, okay, cryonics at the beginning, cryonics probably is going to win the prize faster than chemical brain preservation, because why not? But I knew as a neuroscientist that that from a laboratory technique of looking at neural circuits, that there were these other chemical fixation and plastic embedding techniques that were the gold standard in neuroscience research. And so the Brain Preservation Foundation, the core of it is saying simply that the brain is the structure of the brain, the structure of the neural circuits in the brain, is containing the information that encodes our unique identity. Okay, if there's anything that we can think of is lost fundamentally at our death, it's all of that uniqueness that we have built up over our lifetime. And so instead of letting that brain decay in the grave or be cremated or something like that, it makes sense to have some method, cryonics, chemical fixation, plastic embedding, or something else, some method to preserve what science is telling us is the encoding of who we are, so that future generations can bring us back, or can at least recover some of our uniqueness from our memories, etc. So the core mission of the Brain Preservation Foundation is just to advocate for scientists and medical doctors to work on a technique that could be applied to human patients to preserve their brain so that they can get into the future. Now, we have the main vehicle for us to do this, because the Brain Preservation Foundation does not, it's not a big organization. It is a few people that are all volunteers, that are, there is no laboratory facility or anything like that in the Brain Preservation Foundation. Our main vehicle is the Brain Preservation Prize. Okay, it is a $100,000 prize that was donated by an anonymous donor, that is for whatever scientific researcher, whatever medical doctor can show that an entire large mammalian brain can be preserved in a static state, so that all of the synaptic connections remain intact, and can be demonstrated, skeptically, scientifically, rigorously demonstrated to have preserved that neural connectivity. So that is the Brain Preservation Prize, and there's two phases of it. There's a phase for a small mammal like a mouse, and then the entire prize is for a large mammal, probably a pig or a monkey or something like that. There are two competitor teams currently competing for this prize. One of them is 21st Century Medicine, which is the probably the best research lab for cryopreservation of organs in the world. So they are absolutely a cryopreservation technique lab, and they are working tirelessly at trying to not only do their regular stuff, which is trying to develop a way to preserve an entire kidney for human organ transplantation, which would be really cool, but also to demonstrate that their techniques can be applied to human brains, or to at least animal brains, and to win our prize. We're very excited to have them to be competing in this prize, and they are showing us some very interesting data. Doesn't meet the requirements yet, but that's par for the course. It is a contest. It is a very difficult task. The other group is a group led by Sean McCullough out of Germany, and he is actually the guy who's developing the techniques to preserve an entire mouse brain by chemical preservation and plastic embedding, so that Winfried Dink's multi-beam Sierra-Blockface machine can image the whole thing at a resolution to trace everything. So he has a very scientific goal that he needs to get a whole mouse brain preserved at this level so that he can do his experiments. And he's also interested in seeing if this can be extended to a whole monkey brain. Because neuroscience would really love to get the whole connectome of a monkey one day. So where in all of this is the Brain Preservation's bias, the Brain Preservation Foundation's bias toward chemical fixation and plastic embedding? Well, I'll tell you what it is. It's that we have put them on an equal footing. We've put them on a completely equal footing, and a lot of people don't like them being on a completely equal footing because cryonics, the thought is, cryonics, if you're preserved via cryonics, that somehow you can just be rewarmed and come back in the same body. And that's just medical biostasis, and there's nothing fancy about that. But this chemical fixation and plastic embedding, that requires uploading, and that brings in all sorts of philosophical stuff that people don't want to deal with. And the idea that we put these on equal footing, I think, is causing in a lot of people's minds that we're really radical out there, and that we're advocating chemical preservation and plastic embedding. And that's not the case. We're just not. The Brain Preservation Foundation is not. We're looking at both of these technologies and saying, if either of these technologies can show that you can preserve a brain, this is something we need to get into hospitals. Once it reaches that proven, scientifically rigorous demonstration, then we need to get this into hospitals. And if it was a cryopreservation technique, I would be the happiest that I could be. If it was a chemical fixation technique, I would be the happiest that I could be. It could be that it's a hybrid between the two. There are a lot of work in neuroscience labs where you do a little bit of fixation first and then go to cryopreservation. And actually that gives some of the best tissue quality out there in the neuroscience literature. It's actually what people do for fly brains. So the idea that we're biased in some way, I think, is really it's floating out there, but it's not the case.
Hmm. So, do you think there'd be much difference in the quality of the type, the quality of the self-determination that would come from a mind that was preserved cryonically or chemically via plastination? Like if you tried to get all the data that you could from a cryonically preserved patient and compare that with the data that you could get from a plastinated
Yes.
mine, would there be much difference?
That is a great question, and that is exactly the types of questions that we should be asking when we're comparing these two
Thanks.
technologies. Okay, it is exactly the type of question, and what is really interesting about it is that you see advantages and disadvantages on both sides. Okay, people that are in one camp or the other usually only see their advantages and not their disadvantages, and vice versa, right?
Mm,
So let me clearly spell out the advantages and disadvantages of both. Let's take cryo first. So cryopreservation, the key advantage to cryopreservation is that we can show, people can show on small scales that you can retain some viability. You know, if you're cryopreserving something the size of a human embryo, then you can actually implant it. You can warm it up, implant it, and it comes back as a full-grown human being. You know, so obviously there's a level of chemical preservation that is potentially possible in cryopreservation that would make at least the molecular components of the brain still available if everything goes perfectly well. If everything so if you cryopreserve something, it is potentially possible that you are preserving more of the molecular constituents of synapses, of neurons, neurotransmitters, ion channels, receptor molecules, DNA, all the things that you typically think of. Those are being preserved in such a way via the cold temperature that they can actually come back to life, potentially. But, now here's the bad part, but that works for very small scales. It works for very small scales. And if you try to do that on a large organ or a whole brain, what you have to do is you have to, instead of just lowering the temperature, you have to put in a lot of cryoprotective agent, a lot of antifreeze. And this takes time to put in, and it also causes chemical damage because this is a chemical just like anything else. The goal, so for instance, Alcor, the cryonics, one of the cryonics organization, they pump a very high concentration of cryoprotective agent into the bloodstream of their patients. And they have to do this because if they didn't get a very, very high concentration in, and by high concentration, I mean that most of the water is being extracted out of this. If they didn't get that high concentration in, then when they lowered the temperature, you would get ice crystal formation. And that would cause severe structural damage. And so this causes two things. First of all, extracting the water out of the brain is causing a lot of damage, structural damage and molecular damage. Okay, is it too much? I don't know, but it's causing a lot of change. Okay, and until you actually, so that you can bring a brain back and it's alive, it is very. You should assume that damage, that large amount of visible visual change that you're seeing, may very well be destroying the information that the nervous system has. Secondly, and this is probably even more important, it takes time to get that in. It takes time to get that cryoprotective agent in, and it takes time to get the temperature down low enough that you stop the processes, the metabolic processes that are causing the decay. And so, for the hours that it takes to get a human patient down to a temperature where those processes are stopped because of cold temperature alone, you've got a massive amount of decay processes that are going on. Could that destroy identity? I think it could. We don't know. And the way to figure that out is to look at electron micrographs of cryonically preserved brains and see if the synapses are there, if the connections are there, and how they're disrupted. It's the only way to really determine it, minus bringing an animal back to life, which again has not been done. So those are the disadvantage. The advantage of a cryopreservation technique is, you're not cross-linking. You're not filling with poison and changing the chemical nature of the brain. Okay, but the disadvantages are you're having to take many hours of decay, and you're having to put a lot of this concentrated cryoprotective agent in that is causing structural and molecular damage. Now, those are the advantages and disadvantages of cryo. On the chemical preservation side, the disadvantages are pretty obvious. Pretty obvious. If you inject somebody with glutaraldehyde or formaldehyde, they die really, really fast. This is gumming up just about every protein component of our metabolism, so that it's such a toxic poison that there is just no way you can't. There's no demonstration at the small scale or the medium scale or the large scale that anything can come back from this. The biomolecules are being covalently bonded together. Okay, so this is obviously damage. It's obviously damage to some of the things that constitutes the nervous system. What type of damage is it? Well, it's damage in the sense that the stuff is still there. The receptor molecules are still there. The neurotransmitters are still there. The ion channels are still there. They're locked in place, but they're deformed. The proteins are denatured, and they're cross-linked to each other. So they're in a different state, but the information is still there. That's the disadvantage to it. What is the advantage of chemical preservation? Well, this is the thing that I think that people are not seeing. The advantage of chemical preservation is that within a few seconds, actually maybe a couple of minutes, from the time that you start injecting, perfusing this fixative into somebody's bloodstream, all of the decay processes are stopped dead. It's like you had a ray gun, like in the comics, okay, where you could just freeze somebody instantly. These chemicals that have been used for years in the neuroscience labs, they aggressively go into every cell, and they covalently bond the proteins together, stopping immediately the decay process. And in a way that does not distort, to some extent, it does not distort nearly as much, from what I've seen, the structure as the dehydration process of the cryo does. And so this is why chemical fixation is used for neuroscience research, because if all you're interested in is getting the structure in images, doing your research and mapping out the structure of neural connectivity, then of course injecting the animal with a highly aggressive formaldehyde and glutaraldehyde fixation solution, locking everything in place so that the structure is solid, gives you a tremendous amount of time to then prepare it for other methods and get it into the most pristine state, ready for doing electron microscopy on it. And so these are these two techniques that both have incredible disadvantages and both have incredible advantages. Would you rather have the biomolecules not be covalently bonded together? This may be, from a science perspective, from a science and uploading perspective, this might be the best way of putting it. Would you rather have on cryo? Would you rather have your brain? Would you rather have your brain preserved via cryo, so that the biomolecules are not covalently bonded together, but in order to achieve that, you have severely dehydrated the brain and you have given it several hours of decay? Or would you rather have chemical preservation, where you immediately stop all the things in their tracks, but you do it by covalently bonding all the proteins together? And then once they're covalently bonded together, you can extract the water, and you're not extracting the stuff that was in the water, and you're not structurally changing anything because everything has been bonded together in a nice little matrix. And you can infiltrate it with plastic, and it can sit on the shelf forever. Which one of those would you rather have? And the answer is, I don't know, because neither one of them has been demonstrated to scale to a whole human brain. If I look at a large brain done by cryo, large brain done by chemical preservation, both of them have the problem of not getting the structure of the brain correct currently. And that's why we have a prize. And once somebody wins that prize, I won't be able to say that anymore. And if a cryo wins the prize, it's going to be a cryo. If a chemical wins the prize, it's going to be a chemical. And I can almost guarantee that both of them could win given enough research. And hopefully, once one of them wins, people will see the light and say, oh, we should do it this other way too. Let's have two different ways, and let's have them open. And I would love to see all the hospitals in the world not only be able to provide terminal patients with a better solution than the morgue, I would love to have them have a brain preservation procedure, and the doctor say, would you like the best cryo that's been rigorously demonstrated to preserve the connections of the brain, or would you like the best chemical preservation that's been rigorously demonstrated to preserve the connections of the brain? And then the patient themselves can decide.
That's a great explanation. Now, I think an obvious extension to that question would be, if you were to compare somebody who woke from a slumber to somebody who woke from a sleep, like a cryopreservation suspension or a plastination suspension, what would be the difference, other than just waking up in reality instead of virtual reality? What would be, is there anything that you'd say would be new or missing in an entity that woke up from a plastination suspension, let's call it, or a cryo suspension, compared to someone who just woke up from a long sleep?
Yeah, let me give you a very technical answer to that, if I can.
Okay.
So, from a technical perspective, the conscious processes, from a cognitive science perspective, are based upon symbol manipulation in the brain. It's kind of a vague term, but symbol manipulation in the brain. If when I'm talking about a self model, I'm talking about memories, I'm talking about things like that. These things are discrete entities. I either see a cup or I see a car, right? And these are patterns of neural activity. So if I feel an itch, it's one pattern of neural activity. If I feel a pain in my foot, it's another pattern of neural activity. If I see the color red, it's a pattern of neural activity. If I recall a childhood memory, what I am doing is I am re-invoking patterns of neural activity like I had when I experienced that for the first time, but I am doing it by recalling that based upon synaptic changes in parts of the brain. And so the the thing that explains what this person would feel when they wake up is not at the level of molecules. It's not at the level of neurons. Somebody that wakes up and they've, let's take an example here. Let's say we've got somebody that was cryopreserved, and for whatever reason, nanotechnology has been invented, and the nanobots go in and they repair all of the cryo damage and they put the neurons back into place, and they wake up in the same quote unquote biological body that they died in, that they were preserved in. How would they feel? Well, they would feel the way that they feel, hopefully, like waking up after a long sleep, because those symbolic representations are going off in a particular sequence in their brain. If you had let's say again, a cryopreservation, but the damage level that had occurred, and this may be very likely, that the damage level occurred is just not consistent with the idea of repairing everything with little nanobots, that it really makes no more sense to do that than it would be to drive around Model T Fords, okay? That what actually happens is those cryopreserved brains are scanned in the equivalent of electron microscopes 100 years from now, and it's uploaded into a computer, and now you have a simulation, okay, of that person. And that simulation is connected to a robot body, and that robot body opens up its eyes. What is it going to feel like? It's going to, if any of this worked at all, then it will token the same symbols in the same sequence because of those patterns of neural activity that have been laid down in the original person. And so when that person opens their eyes and says, "Where am I? Who am I? What am I doing here?" They're going to, that process that's going on in their brain, in their mind, is going to shoot neural activity down to the memory centers of the brain. And it's going to say, give me an episodic memory that tells me who I am and tells me what happened and how I got here. And it's going to create the same patterns of neural activity in the simulation. And so the person is going to feel exactly the same. Now, so the answer to your question is you should not be able to tell if you're in a biological body or in a simulated body, or in a simulated brain. I'm sorry, a biological body versus a robotic body. Maybe you'd be able to tell. Okay, you just cut yourself and see if you bleed. But a biological brain versus a computer simulated brain, we should not have any more ability to tell which one is which than we have the ability to tell which neurons we're using to remember our grandmother's face. I don't know which neurons. I could be convinced that I'm not using any neurons to remember my grandmother's face. But that's because my consciousness, my self, resides at an algorithm level that is well insulated from the hardware that it is running on.
So a lot of people think that some gradients of experience come down to the basic physiology of the brain and how it's structured, regardless of the emergent symbols. Right. So what would you say to somebody who said, well, okay, if my neurons were different, regardless of the actual the emergent symbols, okay, then I'd be a different person. So yeah, if my symbols remained the same, exactly the same, but my neural structure changed,
Yeah.
I'd be a different person, right?
Yeah. So,
What would you say to
well,
somebody who suggests this?
yeah. What would I say to them? I would say they're wrong, and here's the reason why they're wrong. Uh, if the symbols are the same, but you feel different, then that means that you would not be able to articulate that difference. You would not be able to. So, for instance, uh, let's say all of the symbol processing in your brain was copied correctly, but the we got one of the neurotransmitters totally wrong, and it gives you a horrible burning pain in your left elbow. Phenomenally.
Hmm.
Okay.
Yeah, yeah.
Uh, would you ever be able to tell anybody about that horrible pain in your left elbow? No, because we got all the symbols correctly, and we know the pathway that gives rise to verbal communication is based upon those symbols. And so we wouldn't be able to convey that. And so it could very well be that if we don't understand some neurotransmitter, that it will cause a completely different symbolic it'll cause a difference in the original brain versus the simulated brain, and the person may wake up with a searing pain in their elbow, but that doesn't mean that the symbols that means that the symbols will be wrong as well. So if we get the symbols right, then and yet there's something different, then that's really treading in the realm of not being a materialist.
Hmm.
It is going against the fundamental core assumptions of cognitive science itself. It could be the case that that's true, but if it is, then basically the neuroscience books and the textbooks and the cognitive science textbooks are just so wrong that it's just a glaring hole. And I don't think anybody should make that assumption lightly. I think the assumption that we should base our reasoning about what will likely happen and what we should do and plan our lives around should be based on the best science that we know, not some gut instinct that, it can't just be symbols. What if it's not using, if it's not using serotonin to do the computations, then maybe it won't feel the same. That has no basis in cognitive science or neuroscience.
Hmm. Hmm. So there doesn't seem to be any direct, like, causation other than like what I'll define as the emergent properties of the brain to consciousness. So the symbolism that emerges from the physical structure is is what we is who we are really. There's no direct one-to-one correlation between any physical structure and the feeling the feeling of consciousness or the phenomenal feeling that we have or the raw feels that we have.
I mean,
Um,
there's definitely a direct connection.
yeah, yeah.
There,
Well, what I
there's
mean
yeah.
is the direct connection. What I'm saying is not determined like as if you're looking at two objects, there's one object at the neural level, at the physical structure level, and there's another object at the consciousness level. There's not one line going directly from one to the other.
Uh, I'm not sure if I understand, but
Sure.
the best way to think about this, um, in my humble opinion,
Yeah.
the best way to think about this is just to be straightforward. Okay, we can't talk about these questions if we don't have a theory of consciousness. Okay, we can you can be in two situations with respect to that question. You can say consciousness is a mystery, We have no idea what it is, no clue whatsoever, and end of story. You know, you can say that, and then but let's not. If that's the opinion, and there's a lot of people that have that opinion. If that's the opinion, then we don't need to dress it up in terms of molecules and neurons and biology and stuff like that. It's just we don't have a clue of what consciousness is, and therefore all of this junk about mind uploading needs to be just tossed out, because we have so much of a non-clue on this issue. We have to pretty much preserve the original body and just hope for the best, because we really don't know what magic goes on. If that's the opinion, that's fine, but you have to understand that if you have that opinion, you're throwing away 100 years of neuroscience and cognitive science knowledge. Now, if you want to take the scientific view of this, then you have to say, okay, well, what is our best theory of consciousness? You know, what is the cognitive science and neuroscience telling us about this? And the cognitive science and neuroscience are telling us that consciousness is a self-model. It is tied to the brain's information processing that models the internal actions and beliefs and memories as something in itself. It's modeling it inside the brain processes. There can be memories that say there can be memories in the brain that say there is a cup on that table. That's not a memory about the self. That's a memory about the external world, right? There can also be memories about the self. I decided to go to Europe last year because I wanted to see the Eiffel Tower. Okay, this is about, I didn't, but this is about the self. This is a piece of information in the brain recording about the self. And we know that the brain does this, obviously, and there are very good theories of consciousness that show how a self-model like this can start to explain all the things that we experience in consciousness, the pains, the perceptions, all of these things, all of these beliefs about who creates our actions. It starts to explain all of these, and much more importantly, it starts to make predictions that turn out to be correct. Just like any good scientific theory, it makes predictions that turn out to be correct. And so these theories of consciousness explain the neurological disorders that we see that other theories never would have guessed would have happened, like split brain patients. It explains why split brain patients feel unified most of the time, and yet they can also behave under other circumstances as having two consciousnesses, and 100 other examples. I definitely recommend Thomas Metzinger's books, because he goes in a few chapters into neurological disorders that really get to the heart. If you want to understand these neurological disorders, this theory of consciousness, this self-model theory of consciousness, explains those in a way that none of these others could explain. And so we should just be honest about are we going to dismiss what neuroscience and cognitive science has been telling us about the self, or are we going to start to accept it? And if we accept it, we will very clearly see that it's the algorithms that matter. It's not the biology.
Okay. So
I know what's frustrating is that I have spent at least a decade of my life reading about this stuff, getting a PhD in neuroscience, and I always feel like I should go into more detail on this.
Yeah.
It's like it sounds vague, I'm just hand waving and that there's a theory of consciousness out there, but there really is a theory of consciousness out there. And
A
popularizers
testable theory.
like Daniel Dennett kind of they fill in that gap of vagueness to a certain extent, where they meet people halfway
Mm,
with their gee, consciousness is really mysterious. I don't see how it could possibly be explained by mere algorithms. And they meet them halfway and show how that gap is really mistaken.
mm, okay. Well, just one more question about, well, actually, maybe one more. But okay, let's just assume that I somebody died and they wanted to preserve the gradient of phenomenal experience that they were having before they died. That was their intention. Um, I've heard, and I haven't really done enough research to see if it's actually the case. Maybe it's just gossip. Is there some sort of membranal sort of charge to neurons or to cells in the brain which disappear after, of course, disappear after the brain is dead and no longer electrically active, that may be needed for that sort of, like, I guess according to what you said before, maybe no, but it's some of the symbols um,
Yeah, I see
requiring this
exactly.
this you do know what exactly I'm talking about? Okay,
I see
you
exactly what
explain
you're
it
saying,
better than
and
I would.
it's a great question, and it could have been that. It could have been that. There is no reason that the brain couldn't have worked that way. So what you're saying is
Hmm.
that uh, what you're really getting to is, can you preserve just the structure
And
of the
without the
neurons
electrical activity, the
without the
real-time activity.
and I can look at parts of my computer that if I were to cut off the power, I would really lose all of the memories that were in there. I would lose them. They would be gone completely.
Yeah,
Nothing would remain in the structure. And I
random
can look at
access
other
memory.
parts of the computer, like the hard drive,
Sure.
and the CPU, and I can say those would still be there. And 100 years ago, if we had asked, uh, does everything that constitutes us, is that really encoded in the structure of the brain, or is there some ongoing electrical activity that, if it was interrupted, would destroy who we are, would lose that information? We wouldn't have known that answer. But today we know it definitively, definitively, okay? Because we've tested it in human beings. Okay, so the way that we've tested this in human beings is by and this is not a test. I mean, they did this in animals long before, but ever since, I think, the late 1950s, maybe 1960s, they've had surgeries called profound hypothermia and circulatory arrest surgeries. Okay, and these are surgeries that are performed on people, usually for brain aneurysms, where the person the aneurysm in the brain that they need to sew up is is too deep, so that they have to operate on the person in a bloodless field. They have to not have blood that's spilling out all over the place, and they need time. They need 30 minutes or an hour of time to actually get in there and do what they need to do. Okay, a lot of aneurysms they can do this without this problem, but at least there are some, and there's some heart surgeries I've heard that needed and maybe still need this type of a surgery, this profound hypothermia and circulatory arrest. And so what they do is they can't just drain somebody's blood at room temperature. Because the the decay that would occur, the damage that would occur at room temperature, if you did that over a 30-minute or an hour operation, would result in complete brain damage. They'd be dead, or they would be nothing left of the person. And so what they started doing is they would cool the person down to somewhere between 15 and 10 degrees Celsius. Okay, this is not freezing. This is not even close to freezing, but it is very low. It's a very low temperature compared to regular metabolism, and it slows the decay process way down so that you can remove the blood from the person. And so you lower the person's temperature down to below 15 degrees C, and you remove the blood. Then you've got 30 minutes or an hour to do your surgery. Then you put the blood back in, restart their heart, and rewarm them. And the person is fine. You can look at the medical literature, and they give case studies of cognitive abilities and memories after this procedure. Now, what is maybe not completely clear in this is that at those temperatures, you can look this up in the literature as well, at those temperatures, the brain's electrical activity is not able to function. They measure this during the operation to know that they've actually gotten to these temperatures. But if you look in animal models, you can show that if you go below maybe 18 degrees Celsius, the neurons are unable to communicate with each other. You've lowered the temperature so much that even if the neurons are still happy and they're metabolizing and everything, the synaptic connections are not working at those low temperatures because of the low temperatures effect on the ion channels that need to open up. And so here we have a case where we can take an adult human being and completely shut down their electrical activity of their brain for 30 minutes to an hour and then reboot them, and they're fine. So the answer is, I can answer with 100% certainty that it is only structure that is needed and not electrical activity. That doesn't answer the question of whether it's the molecules, molecular structure, or the synaptic structure. There's lots of evidence that it really is just the gross synaptic connections and that the molecules are really redundant. Yes, they have to be there to function, but you can, there's research from neuroscientist Kristen Harris that has looked into how memories are formed in the hippocampus, and she does great experiments showing that the changes in synaptic weights that encode memory are easily seen as structural changes. There are molecular changes as well, but they're structural changes, and those structural changes will be preserved in any of these techniques, cryo or chemical.
Excellent. Just quickly, I'll just pause.